cancer in Black women, an immigrant perspective. So let's get started. All right, so breast cancer is the source of a huge health disparity amongst Black women. I mean, here are the facts. Black and African American women are more likely than white women to be diagnosed with breast cancer under the age of 40. And only about half of breast cancers in Black women are diagnosed at a local stage, compared to the 64% in white women. And also, African American women have the highest death rate of all racial and ethnic groups and are 42% more likely to die of breast cancer than white women. So breast cancer is the source of a disparity, and we can attribute that to things like racism and discrimination and lack of access to care. And these could be contributing to the reason why Black women continue to have worse outcomes when it comes to this disease. But what does it mean to be Black in the United States? I mean, the Black population is so diverse. Black people in the US come from everywhere. We speak different languages, we have different cultures, different backgrounds. In the US, 1 out of every 10 Black person is a Black immigrant. And of those immigrants, almost 57% of them either come from Jamaica, Haiti, or Trinidad and Tobago. So there's a huge proportion of them coming from the Caribbean, and they actually outnumber the amount of immigrants that come from Africa to the US. And this is very relevant for Florida because Florida has the second largest Caribbean population in the US, second only to New York. And this is really obvious in where I'm at in Miami, Florida, because we from the map that I have for you here, you can see that Black immigrants form these enclaves. And so there's parts of the community where certain Black immigrants are living in, like Little Haiti that has a lot of Haitians. And in Coconut Grove, there's a lot of Bahamians. And then we also have a historically Black community of Liberty City. And as you can see from the map, they're all close together. So there's really a lot of diversity within the Black population. And sometimes that's not always appreciated when we talk about disparities and health outcomes. And so for that reason, we've turned our attention to Haitians and breast cancer. For one thing, in Miami has the largest population of Haitians outside of Haiti. And so one thing that we looked at, we know that Haitians that immigrate from Haiti and live in Miami, they have better outcomes than Haitians that stay in Haiti. And that can be attributed to things, of course, like lack of access to care and issues with screening and diagnostic tools, the availability of those resources. But as I'm going to show you, that difference is a little bit more nuanced than we think. So looking at this table, you can see that for between the 3 groups of women, Haitians in Haiti, Haitians that are in Miami, and African American women, there are differences and similarities between the presentation of their breast cancer. For one thing, Haitians in Haiti, they tend to get their breast cancer at younger ages, but we see that the Haitians that move to Miami, they're not that far behind. They're pretty close to them. As compared to African American women. But another thing we notice is the type of tumors that develop amongst these women. In ER negative is a type of breast cancer, and we see that it has a lot of prevalence in Haitians in Haiti as compared to Haitians in Miami. And we see that this proportion is more similar between the Haitians in Miami and African American women. So it's showing that at least with types of tumors, it's more similar to US born Black women than it is to the Haitians in Haiti. And we also see that with the stage of cancer, that Haitians in Haiti are diagnosed at a later stage more often. And this trend is more so for Haitians in Miami, is more so geared towards African Americans in Miami than it is to Haitians. So it's going down. And so, but let's take a step back for a moment. And I just wanted to just present the fact that if we look at Caribbean women in general that live in Miami and have breast cancer and compare them to US born Black women with breast cancer, we actually see that Caribbean women have better outcomes than US born Black women. But if we look at Haitian women in particular, If we compare Haitian women with US born Black women and their outcomes, we see that it's actually really similar. And I feel like this highlights a disparity within a disparity, because for some reason, Haitian women in particular are having worse outcomes that is more similar to US born Blacks when compared to women from the Caribbean in general. And so having all of this information kind of gears us towards thinking about, how does immigration have an effect on breast cancer incidence and presentation, particularly in Haitian women? And the way we can do this is that we can investigate the epigenetic differences between women of African descent who have breast cancer and are native to Haiti, the USA, or who have emigrated from Haiti to the USA. And on this slide, I have a picture of the Citadel. It's a very famous fort in Haiti. And I just thought this picture really captures how beautiful the country it is. And I thought we should take a moment to appreciate that. All right, so I mentioned epigenetics. So what is that? Well, epigenetics is what accounts for the changes in our gene expression. So your DNA is your DNA, and your DNA is your DNA everywhere in your body. But epigenetics, we know, can account for which genes get turned on and which genes get turned off. And we know in particular that epigenetics is actually influenced by your environment, and it's influenced by lifestyle choices and factors. So in the case of epigenetics, if you have a gene that is methylated or hypermethylated, which means that methyl groups get added to it, that can lead to the gene being turned off. And you can kind of think about how that could possibly cause cancer for someone. Because if you have a gene whose sole role is to make sure that the cell doesn't turn into cancer, if that gene suddenly gets turned off, then the cell becomes more vulnerable. And it also works on the other side too. So if you have a gene that probably should be turned off because it could cause many problems for the genome, and it's not because it's no longer methylated, so it's hypomethylated, that could also make the cell more vulnerable to becoming cancer. And so we can look at the different methylation profiles of the DNA from an unaffected sample, like the saliva, and the DNA of the tumor, and compare them with each other, and then compare those differences amongst the three groups to see how epigenetics is playing a factor in the presentation of breast cancer between these three groups of women. So that is exactly what we ended up doing. We've collected samples of saliva and tumor from African American women, from Haitian immigrants and Haitian natives. And then we extract the DNA, and through a process called reduced representation by sulfide sequencing that I won't go into, basically we're able to look at the different methylation regions between the two samples, and then compare those groups with each other using computational analysis. And so what we found is that where you live drives breast cancer pathogenesis. So if you look at the green circle is actually representing the different methylation patterns of African Americans and their tumors. And then the blue circle is the Haitian immigrants. And you see that there's more overlap between the Haitian immigrants and the African Americans than there is with the Haitians in Haiti, which is the small circle at the bottom. So their tumors, as far as their methylation regions are concerned, they have more overlap with each other. But there also are many differences that are unique to each group. And so it really just drives the point that if we can understand how these epigenetic factors are influencing what the presentation of the cancer is, and what is the outcome for these three different groups, we can understand more about what causes cancer to form in the first place. And so I just want to end giving some advice about being breast healthy. Be sure to talk to both sides of your family about your family health history, and talk to your doctor about your personal risk for having breast cancer. You can also discuss with your doctor any screening test that might be right for you, and identify if you're at a higher risk. And also, it's been recommended that starting at the age of 40, if you're at average risk, you have a mammogram every year, and just know what's normal for you and make healthy choices, because we know that's influenced by epigenetics is influenced by that. And if you see or feel something, consult a healthcare provider. And with that, I want to say thank you for listening. I want to say thank you to my lab, Alex Sanchez, and also our collaborator in Haiti that helped me out. And with that, I would take any questions.
Thank you so much. That was really informative. I love the breakdown of epigenetics. I have a question though. You mentioned about germline epi, right? Germline epigenetics or methylated DNA. What do you think about, what are your take on somatic DNA methylation?
So yeah, I feel like, yeah, that's definitely something else to look into. Just with the germline DNA, it's easier for us to like compare those differences, because it's almost like the germline DNA is just like a standard to compare it to. But yeah, that's definitely something that's really interesting to look into as well.
Yeah, sorry, I have one more question. I'm sorry. It's not like a deep question, but I know you mentioned people or women in Haiti are diagnosed at a later stage, right? Would you think that's because of an access to healthcare, or what other factors you think would contribute to that?
So I definitely think access to healthcare is a big one, especially because also the lack of infrastructure and not having diagnostic and screening tools in place and protocols so that people at higher risk can be identified earlier on. That's also an issue. So it's definitely an infrastructure issue, but it is interesting to see how even with immigration and being in a country where those resources are, I wouldn't say readily available, but more available than Haiti, definitely changes outcomes a lot. And it just really shows how much, having access to some fundamental tools can really make a big impact on the disease progression.
Thank you. I think we're gonna keep on time. Krystle, you want to keep going?
We had one more question, like from
Okay.
the audience, if we could just ask really quickly. The Christmas asked, what do you think is contributing to the worst outcomes for Haitian women compared to other Caribbean women in general? And if you could also address Jacqueline's question about, could it be a lack of patient education in the States versus immigrant women to get tested? Those two are not necessarily correlated, but if you could address both, that would be great.
Okay, I'll start with the first question. So actually the history of Haitian immigration in Miami is very it's very interesting, because there's definitely been a lot of stigma in Miami around being Haitian. And you can probably trace that to the HIV epidemic, where being Haitian was actually identified as a risk factor for having HIV. And you can only think about the discrimination and the stigmatization of this whole population of people that was just there from the jump. And also, there were policies in place that didn't help, that were like that weren't affecting Haitian immigrants in particular, that didn't help them establish lives here as compared to other groups that immigrated. So I feel like the history of Haitian immigration in Miami definitely plays a role in when we look at comparing the group within other with other Caribbean women. And the second question, I'm sorry, can you repeat the second question for me, please?
Yeah, real quick. They had asked, could it be a lack of patient education in the States for immigrant women to get tested?
Oh, yeah. So that's definitely something that can contribute to it, because we know that having a language barrier can be a huge blockade for someone trying to get healthcare. In Miami, I feel like there is a lot of the Haitian population here, so maybe there are more resources here to address as especially the language barrier. But yes, the lack of education about breast cancer and about what puts you at risk and what lifestyle modifications you can make to put yourself in a better position could definitely be contributing to that.
Thank you so much, Jovanka. We didn't get to everyone else's questions, but Jovanka, if you can like answer their questions in the chat, that would be great. We need to move on though. Thank you so much.
Yeah,
That was so interesting.
thank you.
So next we'll have Krystle Osby, and she's a molecular biology PhD candidate at the University of Utah Huntsman Cancer Institute. And she's in the lab of Dr. Jay Gertz, who I noticed is also here. Okay, support. Today she'll be discussing hormones and uterine cancer. So, Krystle.
Hi, everyone. I'm just sharing my screen. Okay, hi, everybody. Thank you for that introduction. So I'm super excited. Let's see, is it sharing? Is everyone able to see the screen? It's not showing me.
Yes, we can.
Okay, perfect. So I'm super excited to be here to educate you guys all on the role of hormones in uterine cancer, and to also share information on how it disproportionately affects the Black community. So first off, I'm sure not that many people know what uterine cancer is, so I'm just gonna go over the anatomy of this. So uterine cancer is the most common cancer of women's reproductive organs, and it's the fourth most common cancer in women. And here in this image, I'm showing the anatomy of a woman's reproductive system, where we have ovaries, the fallopian tubes, the uterine cavity, the cervix, and the vaginal canal. And most of the uterine cancer cases arise from the endometrium, which refers to the lining of the uterus, which is on the inside here. And now I'm going to compare uterine cancer to other cancers, because I think this is a super important point. There's a lot going on in this chart here, but basically this is summarizing how patients are doing after their cancer diagnosis in the US. Almost all of the arrows are pointing to the right, meaning that patients are living longer after their cancer diagnosis over time. However, the arrow for uterine cancer is actually pointing to the left, and this is indicating that these patients have been doing worse since the 70s. So a woman diagnosed with uterine cancer in the 70s had a better outcome or chance of recovery than a woman diagnosed closer to today. This could be attributed to the fact that we have a limited treatment options for patients. And currently the most effective treatment is women receiving a total hysterectomy, which is basically removal of the uterus and cervix, and often comes along with this is also removal of the ovaries and the fallopian tubes. And when we look at this in more depth with numbers, we see that the number of new cases and deaths are both rising. And in the US, this cancer is diagnosed in 1 in 37 women, and this rate is increasing every single year. This year, it's estimated that there will be more than 66,000 new cases, and about 13,000 women are projected to die of this cancer. And comparing to last year, you can see that these numbers have even increased. Next, I really want to talk about what normally happens in the uterus. So the uterus is, the main role is to prepare for pregnancy or to give women a period, and this is called a menstrual cycle. And there are two main hormones that drive changes that occur during the cycle. And when these hormones work in harmony, things are normal. The hormones estrogen and progesterone fluctuate during different times, and this corresponds to phases of the cycle. In the beginning, you have your period when the uterine lining is shed and bleeding. Then the lining needs to build back up, and estrogen levels rise in this phase, which is called the proliferative phase. And after ovulation, we now have the secretory phase, where we see a progesterone levels rise and help to regulate estrogen levels with drop of it. And during this time, the uterus is releasing chemicals, preparing to either support a pregnancy or to break down the lining again to shed for a period, and this cycle starts over again. And as women reach the end of their reproductive ages, and menopause is considered the end of the cycle, and post-menopause women no longer get a period. But most women with uterine cancer are diagnosed after menopause, and that's almost it's almost always through signs of bleeding after menopause. And this is connected to the idea that hormones are active when they're not supposed to be. So high estrogen levels are a major risk factor for uterine cancer, because it can cause the uterine cells to grow. And this is most commonly seen from obesity. But high estrogen levels are not enough to cause cancer alone. So there must be something else that's contributing to these tumors growing. Almost all uterine tumors contain mutations, which some function of those mutations are unknown, and it's been suggested that they can cooperate with estrogen to cause cancer. So to discuss what I'm researching, my project is asking, how do estrogens cooperate with these mutations to drive a uterine tumor growth? And currently the mutation that I'm looking at is in a protein called beta-catenin. It's a protein that's really important for cell growth. And this mutation allows for this protein to stay active, regardless of signals tell it to turn off. And if both estrogen and beta-catenin are important for cell growth, and they're normally turned off when they're not needed, in the context of this mutation and having excess estrogen in the body, they could be working together to cause cancer. So what I have compared in my research are the differences in the genes expressed between uterine cells in different conditions, where we have cells given lots of estrogen, cells that have active beta-catenin, and cells that have both high levels of estrogen and also active beta-catenin. And this Venn diagram explains how I designed my experiments, where we're really interested in what happens when both of these are active, since we're asking how they cooperate. So we looked at the genes expressed because gene regulation is super important for identifying the program of the cell and predicting a cell's behavior. And gene regulation can control if cells can or grow or die. And this is super important to know when studying cancer. What we learned from my research was that estrogen may be able to control beta-catenin's ability to regulate genes. So we observed that estrogen dominated the system when both estrogen and beta-catenin were active. And after my experiments, the results we found were that genes controlled by estrogen were present. And some of the genes controlled by beta-catenin were present, but some weren't. And also there were new genes that were present, and these genes were not usually active when just estrogen and beta-catenin were individually active. And so what's really exciting is that this new genes were, they might be specific to the cancer growth. And I'm continuing to work on this project to learn more specifics about these genes and what they do and how they're turned on. And hopefully this information can be used to design treatments that may prevent the need for a hysterectomy. And this would completely redesign how we think about helping patients. But this is going to take a while before patients see these results. They have to go through clinical trials and all that, and that takes a long time. So next, what I want to discuss is what's happening now, what's happening today in the clinic. And so something that's super important to note is that uterine cancer disproportionately affects Black women. And this is super surprising because they are actually not more likely to get uterine cancer at all. But when they do, they have a significantly worse outcome. Black women are 2 times more likely to die from uterine cancer. And there have been studies that looked into the cause of this, and it's been found that almost half of Black women receive a diagnosis later. And this is important because later stage diagnoses contribute to worse outcomes. Since I talked before about hysterectomies, just removing the organ, are the most effective as of now, and they're really effective in early stages when this cancer is caught early. So it's very important for patients to get treated as early as possible. On average, almost 70% of uterine cancers are diagnosed early, but we know that the disparity exists, and this is much lower for Black women. The survival rate for uterine cancer caught and treated early is actually 95%. And here you can see the dramatic reduction in survival when it's caught later, which is affecting Black women. So one way we can dramatically reduce this disparity is to receive diagnoses earlier. And the best way to receive an early diagnosis is reporting if you have postmenopausal bleeding to your doctor immediately, and really be relentless that you would like testing for uterine cancer. And so this PSA is really targeted to encourage and empower Black women to be vigilant and to push early diagnoses to help reduce these disparities, because it's no secret that historically we see Black women have not gotten equality in healthcare by having symptoms and like their pain really not being taken seriously by their doctors. So I hope that this PSA and the information that I have explained today radiates through the community and more Black women get early diagnoses. So that's all I have for today. So what I want to do now is just thank everyone who's helped me along the way. I want to give acknowledgments to my lab and my mentor and my thesis committee, because science is super collaborative. And also, if you guys want to know any specifics about my project, I really try to make it super simple, but I use a lot of genomics techniques. So if there's anyone in the audience that wants to know anything more specific about what I do on a day-to-day basis, specific about some of the results I have, feel free to contact me here at my email address. Also, you can find me on Twitter at Krystleography. So thanks everyone for your attention, and I'm happy to answer any questions.
Thank you so much, Krystle. You have a question here in the chat. Sorry, this person is asking, have you ever looked into the sirtuin family, specifically SIRT1, which have been shown to regulate both beta-catenin and aromatase or estrogen production? So this is more a little science heavy for, I guess, for our science community. So apologies in advance.
Yeah, sure. That's a great question. I haven't looked into that as of now, but I am looking into other factors that can regulate beta-catenin and estrogen as well, such as progesterone receptor. So that's something that I'm looking into now, but there's definitely a list of candidates, and I'm just looking through them to see which ones are more probable to be interacting in this model.
Thank you. Okay, so for the sake of time, I don't see any other questions, just praise about your presentation. So for the sake of time, we're gonna move on to Dr. Janae Sweeney. The recently defended Dr. Janae Sweeney is a student from, well, almost not a student, from the Department of Biological Sciences and the Center of Cancer Research and Therapeutic Development at Clark Atlanta University. And she's going to talk today. Sorry, if everyone could please mute themselves. Thanks. Okay, so Dr. Janae is going to be talking today about the functions of antioxidants in prostate cancer.
Thank you, Krystle, for that introduction. Can everyone hear me well?
Yes.
Okay, thank you. Okay, so first I'd like to thank Black in Cancer for allowing me the opportunity to talk about my research today. And as Krystle said, this talk is based on my dissertation research from the laboratory of Dr. Valerie O'Dare-Amara at Clark Atlanta University. And the title is The Novel Roles for Endogenous Antioxidant Enzymes in Prostate Cancer. So just a bit about prostate and the prostate cancer. The prostate is a small walnut-shaped gland that's located just below the bladder and in front of the rectum that functions in producing a fluid that nourishes sperm. And in terms of prostate cancer is caused by an abnormal growth of prostate cancer cells within the prostate, and it's characterized by an abnormal levels of prostate-specific antigen, which is a protein that's secreted by the prostate within the blood. So diagnosed via biopsy, prostate cancer is the most common non-skin cancer and the second leading cause of death among men in the United States. So in 2020 alone, there are an estimated 248,530 estimated new cases of prostate cancer that may be diagnosed, and an estimated 34,130 deaths that may occur due to prostate cancer. And finally, like any cancer, there are several risk factors, including age, family history, race, and lifestyle. And of these risk factors, about 6 in 10 cases of prostate cancer has been shown to occur in men that are over the age of 65. And unfortunately, African Americans have been shown to be more likely to develop prostate cancer, and unfortunately twice as likely to die from the disease. So my research was based on an antioxidant that's found, or antioxidant enzyme that was found in the body, known as manganese superoxide dismutase, or as you'll see me refer to it, SOD2. So the function of this enzyme is to convert any reactive oxygen species within our body that's toxic to our bodies, and from superoxide to less toxic hydrogen peroxide and oxygen. So it's primarily located within our mitochondria, and it's been implicated in several different cancers and diseases, and is known to be inducible by several cancers, by several factors such as cigarette smoke and reactive oxygen species, which, as I said, play a role in creating oxidative stress in the body, which can ultimately lead to cancer. So in my research, I specifically focused on a single nucleotide polymorphism known as a SNP, which occurs when there's a substitution of a single nucleotide at a specific position within our genome. So pretty much it's a mutation that occurs at a specific position within our genome. So here the SOD2 is known to encode for either alanine or valine at the nucleotide 47 position of its sequence. So there have been several studies that have been done that examine the frequency of this mutation among populations in the world. And it's shown that these SNPs exist almost equally among the studied population, and those populations are listed here. So when we think about metastasis of cancer cells, the process that facilitates this metastasis is known as epithelial mesenchymal transition, or EMT. So specifically, EMT is a biochemical process that facilitates the conversion of cube-shaped epithelial cells into motile spindle-shaped mesenchymal cells. So these spindle morphology of these cells is associated now with enhanced invasiveness, migration, and movement of cells. And as I mentioned before, that helps to facilitate cancer metastasis. So finally, the last portion of my research involved muscadine grape skin extract, or MSKE. So the muscadine grape is a grape that is found in wine and is native to southeastern United States. So they contain multiple health benefits such as antioxidant, anti-cancer, and anti-inflammatory properties. And these are things that you can find at the farmer's market. My aunt grows them in her backyard. Muscadine grapes are very accessible. So in research, MSKE has been shown, the extract that is made from the skin of these grapes has been shown to antagonize harmful superoxide species and inhibit prostate cancer cell growth, which promotes apoptosis or cell death. So just getting into some of the data of my research, based on my review of the SOD2 SNP in research, I hypothesized that increased dismutase activity of the SOD2 alanine SNP may inadvertently increase oxidative stress associated with EMT, and it can potentially be antagonized by MSKE in prostate cancer. So I had some main objectives for the study. I'm just going to go through a few for the sake of time. So some of those objectives were my specific aim: one, to create and characterize cells that were overexpressing SOD2 SNPs. And then my aim two, I would investigate the role of SOD2 SNPs in regulating EMT. And again, remember that that's referring to like cancer metastasis. So in the first experiment that we did in this study, we wanted to establish cell models in order to use for our research and create our SOD2 SNPs, or the alanine and valine variants. So my lab advisor purchased a commercially available valine SOD2 SNP, and I used 3 different concentrations to conduct an experiment called site-directed mutagenesis. And this experiment would allow me to take the one SNP, the valine SNP that had this GTT, and again, it had a thymine here, or a T at this location, and mutate it into alanine, which had GCT, or a C at this location where the T is. So here I used the 3 different concentrations, and I did the site-directed mutagenesis. And when we did that, we observed that at the 50 nanogram concentration, the SOD2 was sufficient to cause a mutation. And now I was equipped with both of the variants or mutations that I needed to do my experimentation. So the next portion of the study involved taking those mutations or those variants and transfecting them or overexpressing them into our cells. So integrating them into the DNA of our cells so that our cells can assume these different polymorphisms that we generated. So now that I had everything that I needed, or the variants for these cells inside the cells, I examined what effect they might have on EMT or metastasis. So I found that our alanine SOD2 variants showed more increased activity than our valine variants, which was consistent with what we read about in research. So there's more epithelial mesenchymal transition, or there's more potential for metastasis in these cells now. So here we also determined that the alanine SOD2 SNP cells were also shown to exhibit increased cell growth and migration, both characteristics of EMT. So here I determined that alanine SOD2 may play a potential role in the induction of EMT, and overexpression with our SNP may be associated with increased cell proliferation and migration. So following this observation, I wondered what might happen if our cells that were overexpressed with this internal antioxidant were treated with the MSKE antioxidant that you can find anywhere in nature. So we wanted to determine whether the interaction of these two antioxidants within the cells would actually slow the EMT or slow that potential for metastasis. So I treated the cells with ethanol as a control, hydrogen peroxide, MSKE, and a combination treatment of hydrogen peroxide and MSKE. And what's interesting is that in many therapeutics that are used to treat cancer, they generate hydrogen peroxide within the body. So this combination treatment that we had with our hydrogen peroxide and MSKE, they kind of served as something so that we could examine how patients that are getting chemotherapies might respond to treatment with antioxidants that are found in nature. So here we did a Western blot experiment, and we saw that the MSKE treatment decreased snail and vimentin, as you see here on the left, which are markers of those invasive mesenchymal morphologies, and increased ecadherin, which are markers of the epithelial and less migratory morphologies. So it was kind of reverting those cells from those metastatic characteristics back into more docile epithelial characteristics. So finally, to close the study, I examined how treatment with MSKE and the other treatments I mentioned were affected. So as a reminder, the optimal result here would be that it would decrease cell growth, migration, and apoptosis, which would promote that cell death. So unfortunately, when we looked at cell proliferation or cell growth, it was shown that the treatment with MSKE really didn't affect cell growth negatively or positively. But instead, when we treated with hydrogen peroxide alone and in combination with MSKE, it caused significant decreases in cell growth. So the same trend was seen in cell migration, where treatment with hydrogen peroxide alone and in combination decreased migration. And surprisingly, when we treated our alanine SOD2 cells with our MSKE, migration actually increased, which we didn't expect. And finally, we looked at apoptosis, again, which is cell death. So increases here meant that the cells were dying, which in theory should decrease metastasis. So the same trend as before was also seen here. The MSKE, or the muscadine grape skin extract, wasn't killing the cells. However, the treatment with hydrogen peroxide alone and in combination with MSKE was. So I determined that the cells overexpressed with our antioxidant enzyme benefited more from treatment with hydrogen peroxide alone or in combination with MSKE. So essentially, those patients that are receiving chemotherapies and that are generating hydrogen peroxide within their body may benefit more from simultaneous treatment with an antioxidant such as MSKE. So this is just a graphical conclusion for you to sum up my study. So I treated the cells with our antioxidant enzyme variants of SOD2 and observed increased cell proliferation and induction of EMT in those cells that were overexpressed with our variants. However, when we treated the cells with MSKE alone, it showed no change in cell growth, an increase in cell migration, and a decrease or no change at all in apoptosis. And surprisingly, when those cells were treated here with hydrogen peroxide alone and in combination with MSKE, there were decreases in cell growth, migration, and increases in cell death. So again, this combination treatment with maybe chemotherapeutics and antioxidants may be more beneficial to patients. And finally, I'd like to acknowledge my school and the people who have been instrumental in this research. So my advisor, Dr. Valerie Odero-Mara, and her lab, as well as her former lab members, the institution, Clark Atlanta University, and the Center for Cancer Research and Therapeutic Development, which is where I work and where my research is housed, my collaborator, Dr. Channing Powler at Johns Hopkins University, and my funding through NIH. Thank you.
Thank you so much, Janae. Does anyone have any questions for Janae? We have 2 minutes.
I have a question. Thank you, Dr. Sweeney, for that presentation. What I wanted to ask, so 2 parts to the question, right? So in African-American men, we know that, Black men are diagnosed sometimes at the same stage as a Caucasian counterpart, but they have a worse prognosis, right? And so when we think about clinical trials and we think about Black people being enrolled in these trials, for instance, the drug olaparib, this came out last year, FDA approved, right? For castration-resistant NPC or metastatic castration, prostate cancer. But the trial was maybe 2% Black, not even that, right? And so now we have this drug out that's supposed to help people who develop this type of cancer, and we develop it as a community at a higher rate. So what do you think needs to be done to increase our representation in these clinical trials that are important for that particular type of cancer? And can you also explain for the people who aren't familiar with like the terms metastatic and stuff like that? So number one, could you explain metastasis in relation to prostate cancer? And number two, how do you feel about the representation of Black men in these trials?
So I'll answer the clinical trial portion first. So it is very low. And I think a lot of that entails community awareness. We're not aware of certain things as far as when we should be getting screened, what to actually look for, what are the symptoms of these types of cancers so that we know. And I actually have a family friend who had BPH, which is not prostate cancer, but you see a lot of the same symptoms as prostate cancer. And he just didn't know where to go, what to do. So I think a lot of the things, especially in our community, it starts there. We have to, have forums like this where we can talk in our community about getting screened and things of that nature. And then, maybe we'll have more participation in these clinical trials. And as far as metastasis, so in prostate cancer, your prostate cancer starts in stage 1 within the prostate. And then as it progresses from stage 1 to 2, 3, and 4, it can metastasize to or spread to different parts of the body. So it can go from the prostate and it can spread to the bladder, it can spread to the lungs, the liver, all different parts of the body that are different from where the cancer originally started.
Thank you for that. I think it's also important to know that you stated the prostate cancer spreads to different parts of the body, but most times prostate cancer is, not cured, right? But you can live with it if it's caught early. I think most times
it's very survivable.
Right. But we just miss that opportunity to screen the people in our, in our surrounding, right?
That's one of the reasons why African Americans, unfortunately, we die more from it, because we're not aware. It's diagnosed at later stages when we could have caught it a little bit earlier, and it's very survivable. I know plenty of people who have survived with prostate cancer, but it's all about knowing, that you have prostate cancer in the first place.
Thank you.
Thank you so much, Dr. Janae. We're running out of time. You have 2 questions in the chat. If you can address them in the chat, that would be great.
Okay, no problem. Thank you all.
So we're gonna move on to our last research speaker. Last but certainly not least, my namesake, Bourne. He is a PhD candidate at Weill Cornell or Sloan Kettering Institute, and he's going to discuss today about how we can bring equity to this new groundbreaking research of immunotherapy. So, Chris.
All right, thanks, Krystle, for the introduction. I'm excited to share my work in bringing equity to cutting-edge immunotherapy research. So just an agenda for my talk. First, I'll just give a brief technical overview of the immune system and what immunotherapy is. But then I'll be mainly focusing on my efforts to improve racial inclusion and belonging in this field. So how does the immune system work? How do we fight infections? Well, immune cells are white blood cells that can find infections in the body and eliminate them. And so that's what's illustrated here in this video. I'm gonna wait for it to restart. Okay, so here we have a large white blood cell. Here we have a bacteria and then a bunch of red cells around. And this is how we fight infections. These white blood cells essentially chase, find, and eliminate the bacteria or any other pathogen that can enter our body. And so the overarching question of my research is, what if we could redirect these same immune cells to find and eliminate cancer cells? So in essence, the way that this works is that you have a cancer cell here, and it has a cancer cell marker. And this marker is a protein that distinguishes this cancer cell from other healthy tissue. So the therapy that I study removes some of the white blood cells from the patients. We then use genetic engineering, and we can give these cells a new recognition protein. We just essentially add it to these cells. And this recognition protein recognizes those cancer markers. And then we put these cells back into the patient. And they go, these modified cells go, they find the cancer cell. And when they find them, the immune cell undergoes activation, and then it kills the cancer cell. And so this therapy is called chimeric antigen receptor, or CAR T-cell therapy. And CAR T-cell therapy has seen, amazing results in the clinic. So in certain lymphomas, you can get remission rates as high as 80%. So that means that 80% of patients respond, and you can even get cure rates as high as 40 to 50%. Now, a lot of research is needed to apply this technology to other types of cancer, as well as to improve its work on lymphoma. But I just wanted to provide an example here of how effective this therapy can be. So on the left, this is the patient before treatment, and all of the Black regions represent tumor. And then additionally, what you'll see, the gray regions outlining the skeleton, this is tumor in the bone. So this patient has a lot of tumor everywhere. After the treatment, what you see on the right is that many of these Black regions are completely gone, as well as the tumor in the bone. And a lot of the remaining signal that you see, the remaining Black regions, these are just organs that tend to light up in these types of diagnostics. So this patient was essentially cured. So now, if we want to further improve CAR T-cell therapy and other immunotherapies, we have to go through a very lengthy approval process. So, this drug development pipeline here, where we begin at drug discovery, then we test them in preclinical models, and then we go into clinical trials and eventually approval. And as with many other therapeutics, Black scientists, physicians, and clinical trial participants are underrepresented at each and every step of this process. And to make matters even worse, a cutting-edge therapy like CAR T-cells can cost about $500,000. So that provides a substantial barrier. So in my work, I focus mainly on these first two areas of the drug development pipeline, which is drug discovery and the preclinical models, essentially trying to improve upon CAR T-cells and expand them to other tumor types. But rather than focus on the technical details of this work, I want to discuss the opportunities that I've had in graduate school to improve representation in this field. And so that's what I'll be addressing in the next part of the talk. And so the first, obviously, is Black in Cancer. Here we are. Black in Cancer was started just this summer, and it's in response to the very poor representation in cancer research. So on a personal level, at my graduate institute, of over 200, maybe even 250 total professors, only one identifies as Black. That's a very stark disparity. And the question is, well, how do we address this disparity? And I think Black in Cancer definitely provides an answer. So two of our pillars just going forward, and I know Henry mentioned this in the beginning, but we're focused on community education, such as this talk that I'm giving right now. And we're also focused on mentorship for early career scientists. And the opportunities with Black in Cancer have just begun. I don't want to give any spoilers, but we have a lot of opportunities, events, and things down the pipeline for 2021, and I'm very excited. Another opportunity that I've had to contribute to is summer mentorship programs for underrepresented students. So at my graduate institution, we have a program called the Access Program, and essentially we target underrepresented students. And we're part of this consortium called the Leadership Alliance. So this is a group of institutions of higher education, higher learning that are committed to these types of programs, bringing in underrepresented students and training them to be the scientists of the future. And this point is very important. We pay our students. So we bring them in for these research opportunities. We provide them with a stipend. And I know that when I was an undergraduate student, all of the money that I made over the summer would, sustain me throughout the entire year. So it's very important to pay students so that we don't put financial barriers to access in science. And then we give very valuable career and skill development. So not only are these students all in the laboratory doing research, but they're also learning how to write, how to read scientific papers, how to interview for graduate school programs, how to apply for graduate school programs. And of course, many of these students go to graduate school and medical school. So, just in my experience, one of the students that I mentored is actually interviewing at Weill Cornell right now, like as I'm giving this talk. Also, another student that was, in the lab, he's now an MD PhD student at Northwestern. And many of the students that you see in this photo have come back to Weill Cornell as research technicians or graduate students. And I think very importantly, these types of programs boost confidence and combat imposter syndrome because we provide a community for students where they feel like they belong in the scientific community. And we talk about issues of racial identity. And there are many other similar programs. So for any aspiring scientists, or if you know an aspiring scientist, the Weill Cornell Access Program isn't the only one. There are many that are part of the Leadership Alliance Consortium, and they work. They're very effective. And then lastly, I want to talk about protest and organizing. So this past summer, as the Black Lives Matter protests really began to ramp up, as I mentioned, my graduate institution has a severe problem with diversity, especially in leadership positions. And so I organized petitions with other diversity, equity, and inclusion groups and clubs to address the structural racism within our own research community. And they listened. And so as a result, we formed a task force of about 30 to 40 scholars. And this ranges from postdocs, professors and staff to technicians, graduate students, pretty much everybody. We wanted not only a racial diversity, but also a diversity in terms of hierarchy. And then on this task force, we report directly to graduate school leadership so that we can hold them accountable and they can also hold us accountable for making this progress. And so we're making very incremental but tangible progress at this point. One of the recent victories is that we secured funding for a summer research program that targets underrepresented students that have not yet had a laboratory experience. So this is widening the pipeline of students. And so just to conclude, I want to address the question of why do we need more inclusion in biomedical research? Or sometimes the way that I even phrase this question to myself is, why should I dedicate myself to these efforts despite losing research time? And so the first is that obviously it improves health in marginalized communities. So I'm in the business of improving health, and having Black researchers in these positions of power, we think about diseases that impact the Black community, as you've seen from the other talks. We also think about including Black patient samples in our work. Secondly, it captures more brilliant scientists, which improves scientific quality. So one of the things that I've noticed is that underrepresented students, particularly Black students, have such exceptional potential due to lived experiences. And in my own research, I've been fortunate to work with a number of Black students, and each and every time, the quality of my science is vastly and dramatically improved. So there's a pool of brilliant scientists out there, and we just have to foster their progression. And then lastly, it fosters trust and accountability in research. So it's very important that our Black community sees our scientific community as us rather than them. And that's how we can get trust from the community and improve health. And so just to conclude, cancer immunotherapy, CAR T-cell therapy is a cutting edge cancer treatment, and it's saving lives. And I'm very excited to be able to contribute to this research. And Black researchers are advancing the research in this field, but we are vastly underrepresented. And so the initiatives like Black in Cancer, the mentorship programs, funding opportunities, and organized protests certainly work. They move the needle on diversity and representation. And so with that, I'd just like to acknowledge all the groups that have been instrumental in my graduate career so far: Black in Cancer, the Scheinberg Lab at Memorial Sloan Kettering Cancer Center, the Weill Cornell Access Program, as well as the Weill Cornell Social Justice and Anti-Racism Task Force. And with that, I'll take questions. Thank you.
Chris, thank you so much for that. And I think that's going to be like the perfect segue into our keynote talk. Does anyone have any specific questions for Chris? In the meantime, I do have a question. Do you have like ideas, I guess, of how we can get started with introducing cancer research to the K through 12 students rather than just, undergrads? I feel like a lot of programs right now are catered towards undergraduate students and introducing them there. But I do know, like, at MSK specifically, there is like a high school program, which I think is excellent for trying to introduce cancer research or cancer biology to high school students. But how do we introduce it as early as even pre-K or K? Like, how do we start teaching them about cancer research?
Yeah, that's an excellent question, and it's one that I'm thinking about actively. Right now, one of the things that I'm actually working on is a vaccine hesitancy program where we go into high schools. And I think one of the key things that we talked about among our group as we plan this is representation. So when we go to the high school, the presenter, the lecturer, whoever the case may be, needs to be representative of that community. Again, this idea of us versus them. And so I think it's just, seeing what you can become. I think it's as simple as, being a Black person and going into the Black community, into a high school or even middle school and earlier and saying, I'm a scientist, you can be one too. And now in the world of, everything being virtual, I think we're probably less constrained by geographical issues. So I think that we could really take advantage of platforms like Zoom that we're on right now to be able to get into much younger stages and just show them you can be a scientist. I'm a scientist, and so can you. You can be one as well.
Thank you so much, Chris.
One quick question for Chris. Is that okay? Yeah, Chris, I was going to ask, is there are you aware of any parallel work in your field going on elsewhere?
Parallel work?
So what I mean is do you know of any team anywhere in the world doing exactly the same thing you're doing on immunotherapy?
Well, yeah, with immunotherapy, there's a ton of institutions that are working on this. CAR T cells, because of the manufacturing process, I think is restricted to even less institutions. So there's only a handful that are really doing a lot of the work to drive this forward, which is actually one of the reasons why access to it is particularly difficult, because it's, some of the largest academic institutions that can do this, and it's not your everyday hospital.
Because in that case, international collaboration might be a way forward, considering how much it costs to do this sort of work. Interesting.
Yeah, absolutely. Collaboration is needed because it has a lot of moving parts. Immunotherapy research and using primary samples, samples that come from patients or humans. Very difficult work that requires a lot of different instrumentation as well.
Okay, with that said, we're going to move on so we can keep time. Thank you so much to all of our research speakers, Jovanka, Krystle, Dr. Janae, and Chris. Thank you guys so much for stepping up and sharing your research and your topics with us. With that, I'll pass it on to Henry.
Oh, sorry, I was on mute. Yeah, it's been great. Love the presentations. Really, really great to see people in our community who are deep in the field doing the research on the front grounds doing this work. It's really important for everyone to see this. So I have the privilege of introducing the keynote speaker, Dr. Kelvin Jones. He and I attended Tuskegee University together in the Integrative Biosciences PhD program. We're in the same lab. And so one of the things I love about Kelvin is that he's able to translate not only the stuff that he does at work, educating the community, but bringing it into a way that we are motivated in everyday life. And so I could not think of a better person to bring Dr. Kelvin Jones, who's also an alumnus of Alabama State University, so double HBCU up in here. And so I really wanted to bring him on to kind of motivate us, because especially at a time like this, where we see so much, violence on TV, our climate in the States you know, we really need to be encouraged to keep going and keep fighting the fight against cancer, because it's affecting our people, our family the most, right? So how do we keep going as academics, as industry professionals, as advocates for cancer in general? And so I would like to present Dr. Kelvin Jones as he motivates us on how to revitalize representation in cancer research and medicine.
Good afternoon. Good afternoon, all. It's a pleasure to be here. Thank you so much. Well, as we continue in what Ms. Gorman described as seemingly, she described in her inaugural poem as a seemingly never-ending shade, I'm grateful for lights. I'm grateful for visionaries like Dr. Henderson and soon to be Dr. Bell. I'm excited about hearing her complete her work. And then even this great team of advocates for the culture through the excellence in science. I'm so grateful that we have phenomenal cheerleaders cultivating an environment of success for those who will one day come after us. I am humbled to be in your presence. Good afternoon. Thank you for the invitation. I'm grateful to be here. I'm grateful to be able to be numbered amongst so many great individuals. I look across my screen and I see awesome people doing great things. And what I'm excited about is they look like me. That's awesome. We're living in an amazing time. Who would have thought 2 years ago, maybe about 18 months ago even, COVID-19 would have impacted the world like it has? Who would have shut down businesses, schools, whole cities, countries even, by a vaccine in this day and age? Who would have thought that would have happened? Very few, I tell you. But in spite of that, who would have thought that in 9 months, with all the red tape and the bureaucracy that goes on in the United States, that we would have been able to design, develop, and get FDA approval for a vaccination for that same, in 9 months? Who would have thought that would have happened? I'm so grateful that we have numbered amongst those who were in the development stage, Dr. Corbett. She looks like us. She looks like you or me. And she was integral in the development of this vaccine. She was there. She was in the room, making sure things were put together right, that we were represented. And so I'm so excited that even through this hardship, there's some goodness there. I feel like I can borrow a line from Charles Dickens. It's the best of times, it's the worst of times, because we're dealing with this negativity, this sickness and this pandemic, but we're also dealing with negativity like hatred and bigotry that's in our governmental, in the governmental spectrum. It's even in our neighbors and those people who we called friends. We didn't know that they may have held certain mindsets. And our colleagues, well, we kind of had an idea, but we weren't that sure. But it's starting to come to light that these people who have such horrible thought processes about us and people that don't look like them are still around. And to boot, we have to couple that with the ongoing health disparities that we face. We are the minority, meaning the amounts of our culture, our population is small in comparison to those that are around us. And even though that's the case, we lead the way in things like cancer mortality. Women and men lead the way in breast cancer or prostate cancer deaths. There is an issue. Just like we had Dr. Corbett in the room when they were designing and developing the vaccine, we need us at the table. We need us in the room when these new therapies are being developed, when they're dealing with new ways to roll out these vaccines. We need us in the room when they're figuring out who's going to get the money, who's going to get the grant, who's going to be the voice or the face that goes out and talks to our communities. We need to be at the table. You know, I grew up in Springfield, Massachusetts, great city. But I remember growing up in that great city, and it was divided. It was a city, blacks stayed with the blacks, and you could see through the city that there was divisions. This is what I grew up in. I grew up. My dad is a construction worker, and my mom was a nurse's assistant. Neither of them finished college. I ended up being a first generation college student. That brought a lot of stress. Those of you that are like me, that's a lot of stress because you have to do what others aren't able to do. You have to figure out how to get your English work done, get your science work done, your math work done. At the same time, sometimes you have to figure out how to handle financial bills and issues that someone before you didn't have to deal with. You have to deal with hurdles of learning how to network properly and how to dress when you go into certain rooms. There are all kinds of hurdles that we have to deal with, and it causes us to feel like, maybe I just shouldn't be here. Maybe I don't belong. I had the pleasure of, right when that started taking hold, to be a part of, as Dr. Henderson so stated, I earned my bachelor's degree in chemistry from the Alabama State University. Very proud to be a mighty marching hornet. I'm so proud to represent my alumni, my alma mater. So proud that I was able to, while I was there, be a part of a research society, the MARC program. And there, I was surrounded by people that had similar mindsets and similar trajectories as myself. We were all going to graduate studies. There was the MARC program, there was the STEM program, there was the MBRS RISE. There were all these programs, and they were all around me. Everybody pushing to get to the same place. We would go to the different banquets, and there would be individuals who got similar GPAs as me or better, all there. And you start to think, wow, they're smart. and maybe I don't belong here. Even though all these things are going on, this is what you start to think, because you think that you're by yourself, and you think that you're the only one dealing with these issues, because no one came to me and said that I too was thinking that I'm not getting this, but I don't want to tell the teacher, so I have to go home and study twice as hard because what he said didn't make sense to me, because the school I went to didn't quite prepare me for this. No one told me about how, while they were going through their studies, they felt like quitting every other week because the money issues that they were dealing with were growing week after week. No one told me these things. And so I was sitting there thinking, I don't belong. Well, I want you to know you're not alone with that thought process. I wasn't alone. You weren't alone. But we pushed, right? We pushed. A lot of us dealt with this issue. A lot of us dealt with this mindset, but we pushed. And I'm happy for you. Those that are still pushing, keep pushing, right? We pushed through our undergraduate careers, and we went on to graduate school. Some of us masters, some of us straight into our doctorates, some of us to medical school and the like. We pushed. And I wish I could say that when I got to graduate school, because I graduated from undergrad, that thought process of not belonging faded. I wish I could say that when I was in graduate school, winning awards at conferences and achieving all these various awards, that I would no longer feel that way. But the sad truth is, I did. And to make it worse, I'll tell you a story. It was about, I think, a year. Henry probably knows better than me, but it was probably a year into I had finished all my classes, and it was time for me to get my research going. And a year into my research, we had finished my first aim. We were heading toward the end of my first aim and moving into the second. So it was like a blurred line there. And it seemed like as soon as I was about to accomplish something great, contamination hit all my cells, contamination hit everything. And I just got so frustrated. I was dealing with research, I was TAing a class, I was doing all these things, trying to make sure that I was being where I was supposed to be and hitting the marks where I was supposed to hit the marks, and making sure that no one thought that I was not as smart as everyone thought that I was. You know, all this was going on. And then everything fell apart. It got so bad for me, guys, that I ended up in the hospital. I was in the hospital for a couple of days because my blood pressure got out of line, and the stress of trying to perform and meet these goals and my attempts to be perfect, because a PhD means you are perfect, right? All that began to overwhelm me, and I just said, you know, I'm going to call it quits. My health is more important. And I went and talked to one of my mentors, and he said, I expected him to be truthful. I went to him because I expected him to talk me out of it and say, keep going. I got there and he told me, he says, oh, what you're doing is difficult. See, what I was trying to do was I was trying to use bacteriophage to isolate a protein on the surface of metastatic cancer cells, right? And so he began to say, imagine how small that must be. That must be very difficult. You need to find something else. You need to do something else, or maybe you need to go another route. And I said, oh my goodness, I came here for encouragement. And instead of being encouraged, I feel more stressed. And so I went and talked to someone else and said, you know what, I was talking with this mentor and they said this, I think I'm gonna call it quits because this is not gonna work. And they said, don't quit, keep pushing. And I said, oh God, here we go. And so I went back, I pushed, right? And I began to look at what I had to do, and I broke down where I had to go and each step that was necessary. And I pushed through, and lo and behold, in spite of all that was going on negative, things began to fall into place, and what wouldn't go right went right. What was going wrong started going right. And before you know it, everything fell into place. And I was able to do what only 6 other labs in the world has done. I was able to not only target a protein on the surface of a cell, I was able to identify that target and open up a whole new line of research into what the reason or why is it present there. It was a great time. You know, I was at an HBCU, Tuskegee University, and we did something great. We were able to patent our research, our peptide, because this peptide had high affinity for metastatic cancer cells. It was great. And so I was excited. I got my PhD. I walked across the stage. I got hooded, and all those things were great. You would think with all these great accomplishments, as I was moving forward, I would start to feel like you belong. But the truth of the matter is, the more I moved forward, the more I felt like I don't belong. I got lucky. I slipped up and made this discovery. Somebody's going to figure out that, you know, he just got lucky. And, you know, I wasn't alone in that thought process. Would you believe it that Maya Angelou, as great as she is, as great of an author that she is, she felt the same way. 11 books into her career, she felt like someone was going to come along and say, you're an imposter. You don't belong here. The imposter syndrome is real. The imposter syndrome is a real issue. And I want you to know that you are not alone. To be truthful, I had earned my degree. I was Dr. Kelvin M. Jones II. And I enjoyed saying it. I enjoyed the fact, I just liked how it sounded. And so I went forward looking for careers, looking for postdocs, got this one, got that one, yay. And I had came across someone who I held in great esteem, great esteem. Their words, their thoughts for me were important. I held their research high. I really appreciated what they did, and so I began to talk with them and share with them, and all the while hoping they don't ask me something I wasn't going to know the answer to, because then they want to think he got this degree, but he doesn't deserve it. He got this degree and he probably doesn't earn it. He's not really a doctor, not like I'm a doctor. You know, that's what I thought. And so we're talking, and one day she said what, I shouldn't have said she. I don't want pointing people in my past, but this person rudely addressed me. And they did what I was fearing all along. They started treating me like, because I made a mistake. I'll let you know. I'm human. I learned that I'm human, and I made a mistake. And they berated me and tore me down to the point where I started believing that I didn't belong. I started believing that I shouldn't be here. I started believing that I accidentally earned my degree. And so in the midst of my despair and depression and wanting to give up and curl into a ball, I had the joy. I have a group of friends who called me because they heard in my voice when they were talking to me that something wasn't right. And these group of friends got together and we started talking. They talked about where I had come from, what I had done, and what I, not really knowing what had gone on, but they began to talk to me, and they began to tell me about how we came from one of the worst streets in Springfield, and you did this, and you achieved this, and we're so proud of you, and you did this. What I want you to know is I had a great group of friends who reminded me not who I was, not what I had done, but who I am. They reminded me that greatness was not in my past, but greatness was who I was. And so on today, many of you, I'm sure, are dealing with some of the same issues that I dealt with. The best thing what I learned to do when dealing with that individual, I learned to take that big situation that was in front of me. And instead of looking at that big situation, I had to break it down into small ones. Small attainable goals. And each time I had a small attainable goal, once I reached that goal, I had a celebration. I don't care if my celebration was nothing more than a slice of pizza and a movie. I had a celebration and I put it on a calendar. My wife, she would do something on Facebook. There would be some kind of point there. There was a demarcation in time to celebrate my accomplishment. And so once I celebrated that accomplishment, I can go to the next one. And what would happen is, after a while, I don't want to fool you and make you feel like that voice of not belonging faded. But every time it would show back up, I had those times where I had celebrated. I had those to fall back on. Remember when you did this? Remember, you came from a lab where you did something only 6 other labs did. Well, 5 other labs did. You did that. And so I had to remind myself how I was able to do something. Sometimes, I don't know if, no, he wasn't doing that, but sometimes you have to pat yourself on the back. You have to remind yourself of the accomplishments that you did, because that imposter syndrome will speak in your ear and tell you that you don't belong, that you shouldn't be here. But I'm here to tell you something different. You graduated, and I don't want to make small of this. You graduated high school and you went to college. There's a lot of people that don't do that. But you didn't just go to college. You went and you graduated from undergrad. Many of us graduated from undergrad, and some of us are still working on it. But then you turned around and went to graduate school and decided to get a PhD or a graduate studies. I want you to know that you have moved from being a large part of society to being an even smaller part of society. I want you to know that you've done something that many people haven't had the heart or the desire to do. You've surpassed the desire to quit, and you went on to be greatness. And so every time that thought comes to your ear, or that voice speaks into your ear saying that you don't belong, remind them of where you came from. Remind them that you achieved, you got your degree, and maybe you just barely got cum laude, or maybe you didn't. Maybe you got summa or cum magna. Whatever you got it. You earned it, right? They didn't give it to you. You earned your degree. You earned your spot where you are. And so don't ever let anyone tell you you don't belong. Instead, surround yourself with people that can help you, that it can encourage you. I made the mistake of thinking that anytime you go to get help is good, right? But not all help is good. You have to make sure you get good help, right? And so unconsciously, I canceled out the voice of that mentor who told me all those negative things about what I should do or shouldn't do as far as it pertained to my research. And I got good help. I got good advice. And that good advice, although it was very simplistic in nature, keep on pushing, was what I needed to hear. Subconsciously, I was looking for someone to tell me to push. And so on today, I'm here to tell you to push. I'm here to tell you to take what you've got. And if it looks insurmountable, if it looks too big, break it down into steps. And if it's only one, if all you can say is, today, my success was getting back in the lab. Take a picture, post it on Facebook, put it on Instagram, and say, look, y'all, I'm in the lab. Take your picture and celebrate that moment. Because what Facebook will do, they'll remind you in a few months, look at this picture from a few months ago. And you'll look and say, wow, I celebrated that. Now look at me. I was able to get this publication. I was able to go to this conference, whether virtual or in person, and I won this award. I was able to do all these things. And each time, make a point of celebrating each achievement. It will help you. It will push you to where you have to go. It will cause you to have the energy necessary to go beyond where you are right now. If Maya Angelou could get past it, if I could get past it, I know you can get past this moment. And so I'm gonna try to bring this to a close. I'm gonna try to wrap this up. I earned my degree from Tuskegee University. I was so happy. I was so happy that I was able to accomplish something so amazing. This allowed me to be able to be in the room where other individuals were helping to decide who would come on to this grant or who would be considered for it to come on to this new group that a particular school was working on. And I got to be an advocate for someone who looked like me. They didn't have the best answers. They didn't have the best individual speaking up for them, but they had a good degree, good grades, and they had good research. They maybe weren't as connected or been able to network with other individuals, but there were strengths to them that needed to be spoken of. And so I got to speak up for them. Today, that was one of my rooms. You know, one of my tables I got to sit at. But today, we need you at your table. We need you at the table. Your table may look different from mine and may look different for the person in the square next to you on this Zoom. Each of us have different tables. You know, some of us are in hospitals caring for patients. That's your table. Others are at the bench conducting research experiments. That's your table. Others are in the field conducting clinical research or teaching or helping to design various therapies and the like. Those are your tables. And I don't want you to diminish yourself. Your table is important. Your seat at the table is important because you being at the table will ensure that someone coming behind you can be heard. Someone coming behind you can be seen. We need you to show up at the table. We need you to be there in the room when individuals are designing these next therapies, because we need more than 2% of the population of the individuals being tested for the vaccine to make sure that it's befitting. Because, you know, I was listening to someone's talk earlier. Forgive me, I'm horrible with names. But to think that 2% is sufficient, no. We need to make sure that we're there, not just designing the drugs, but going out into the communities, informing what this was looking like, who was there designing this, taking away the fear of possibly being harmed when all we want to do is help. We need you at the table. So use every accomplishment that you have to quiet the voice of that imposter syndrome. Quiet the voice of those individuals who think you don't belong there, and take your seat. Take your seat at the table. Sit there proudly, knowing that whatever university or college that you came from, you earned your degree. You earned your spot. That what you're dealing with is particular to you, but it's important because it helps make you who you are. It helps design, bring out those intricacies that you developed when you were growing up. There's parts of me that are key because I grew up in a household of a construction worker and a nurse's assistant. It's parts that are key to me that grew up because I grew up in a lower income neighborhood where I saw individuals get shot and not make it out of middle school. Those things help design or help bring together the various parts of me. And instead of hiding them, we need to celebrate them. Not celebrate the negativity, but celebrate the fact that we were able to come out of it and excel and become the greatness that we are. I want you to know that you're excellent. You're excellent not because of your degree. You're excellent not because of your achievements, but your Black is beautiful and it's excellent. And I want you to know what makes it even more excellent is that you had the nerve to bring that Black beauty to science. And so on today, I celebrate your Black excellence. And I want you to know we need you at the table. We need you in the room. Thank you.
Wow. Yeah, I'm over here. I had to have my camera off. I was in a little praise break. I wanted to throw my shoe at the screen. Wow. Kelvin, thank you so much. You guys can see why I asked him to do this. Many times in the lab, in graduate school, where we both were like fed up with just everything. And so he's the one that got me through, and we got each other through. And so just having him to give this talk, I didn't want it to be focused on the science, right? But how do we keep going at a time where we're feeling imposter syndrome, when we're feeling like we're not enough? And so I really thank you for that. One of the questions I want to ask and pose is that you mentioned that we should seek good help, and that there's a difference between regular help, right, and good help. How do we, scan or figure out if a mentor has our best interest, especially when, as graduate students, they're seeking which lab they're going to spend the next 4 to 5 years. For postdocs, which lab they're going to spend their life in for the next 2 to 5 years. How do we, pick the right people for us that can get us to the point that we want to get? How do we seek good help?
That's a good question. What we have to first learn is that there's a good game that everybody plays. And so people will put forth their best face, right? And they'll tell you exactly what you want to hear. They'll tell you that I'm so excited to have, to be able to help bring someone up and to ensure that there's diversity in science. But if you're constantly talking about my color, when you're talking about me coming into the room, I feel like you just get me for a check. And so you need to be mindful of that. That should be something that goes off, a light that goes off. I'm not saying get rid of them because that's what they're bringing up, but pay attention to that. Then you need to go look at the track record. How many graduate students have they had? How many postdocs have they had? What have they gone on to do? How connected are they with those individuals that left their lab? Is the first thing they have to say is something negative about someone that was just in their lab? My grandmother used to put it like this, and she's not, while she is not collegiately trained, she is very well diverse in knowledge. And she told me, a dog that'll bring a bone will carry one. And what that basically means, if they're gonna talk about you, that person, they're gonna talk about you. And so be mindful of the individual that's going to come to you talking negatively about someone else. Oh, my previous postdoc was horrible, but I'm excited about having you in because I believe you're going to make the changes that they couldn't. If you have to diminish someone else to try to make me look good, that's a problem, because then you'll probably diminish me to make someone else look good. You need to look at not just their funding, but how much compassion that they show, compassion for their lab, the people in their lab, and for the people that they're actually tending to. What is their passion? What's driving them? Is money driving them? Because if money is driving them, once they've gotten all the money they can get for you being in their lab, that help fades. And so you want someone that's concerned with your advancement. You want someone talking about asking you where you see yourself going, and what do you need from them in order to get there. You want someone that is asking you where you want to be in 5 years and what can I do to help you get there. And after they say that, you want to be able to see that they've done that for someone else. If they're new, okay, well, recognize there's going to be a limited amount of stuff that they can do, right? But you want to be making sure that that individual has your best interest in mind over just what you can do for them.
Yeah, thank you for that. I think that's perfect advice, because a lot of people don't know how to navigate those conversations and navigate pinpointing a successful mentor. I think, being a PI and being a mentor are two separate entities, right? Not a lot of people can be both. And some people are good at one, some people are good at the other. And so it's really good to find people who are able to tap into both of those areas. So thank you for that. Another question that was posed into the chat was, how can we as graduate students and beyond foster environments that help Black students to feel a sense of belonging? I know it's a little different at HBCUs because we're surrounded by people who look like us, right? Everybody's, on the same level, or, we all are from the same backgrounds or similar backgrounds. But when people are at different schools that are majority majority white, how do they, foster environments to help them belong amongst their own communities?
Well, I had the privilege of also being a postdoc at a non-minority serving institution, non-HBCU. And what I witnessed there, both in the time when I was there as a undergrad doing an internship, summer internship, and as a postdoc, I recognized that there was a group of individuals that had came together. It was the Black Postdoctoral Association. And what they did was they made sure to put together time, and they sought out individuals who had come to the school and had similar mindset to them. They made sure that when it was time for when you're first coming onto campus, there was someone there to check to see if there was anybody new that might come in and that might be interested in being a part. Help to set up, and especially with this these virtual capabilities, help set up some atmospheres where you can help someone study. Maybe you can set up a study group. Maybe you can set up just a session where you do something that's completely unrelated to science, but reflects, a comfortable space for them to feel comfortable with. Because at the end of the day, sometimes when you leave that lab, you just want to be with someone where you don't have to talk science, where you don't have to, you can let down your guard and a little bit, just let it down a little bit, not too much, but let down your guard and be free for a moment. So if you can set up some moments like that, they will not only allow you to build lasting relationships, but then that same person that you could share a slice of pizza with will be that same person you can talk to about when something's going wrong, when you need that extra supply, when you need that help figuring out how to work this protocol out. They'll be there. And so the best time to build connections is not through the science. They have this saying in business: the best business deals are not brokered at the table and in the business room, I should say, but they're done on the golf course during leisure times. So build up leisure times and give space for those times. Even if you're in a predominantly white institution, build a space, a safe space where you can come together and have fun away from your science. it will allow you to come together and talk about your science later. I can be long-winded, so I'll be quiet.
All right, thank you for that. Another question, how do we overcome failure? I think as scientists, getting frustrated in the lab about an experiment that worked one time and then N2 and N3 fails, and then 3 months down the line, you see the same result. Like, how do we get over that hump, right? How do we get over failure? How do we get over failure in our everyday life, applying to jobs, not getting the job that we thought we wanted? But how do we navigate failure, and how do we not let failure hold us back from achieving our ultimate goals?
When I was in the lab, I had a experiment that I was working on. I'll deal with my undergrad experience. I was dealing with trying to make recombinant phages. And this was something that was pretty difficult. And I went 3 months without seeing a positive clone. 3 months without seeing one positive. What I started doing was giving up because it felt like I couldn't, it wasn't happening. And then what I did was, instead of giving up, I looked at exactly what I had done because I had made sure to write down in my notebook what I had done each time. I started going back and seeing what I could have done slightly different. Fast forward, the same thing happened for me when I was during my PhD. I was looking at my cancer cells and I couldn't figure out why I couldn't get this protein to be isolated off this surface, off the surface of the cell. And one thing I kept noticing is when I would add this reagent, the media would turn slightly yellow. And I didn't pay it attention. Common sense would have told me it now that I look back, but then I wasn't paying it attention. What the cells were trying to tell me was it was an acidic environment. Usually there's something there that will show you where you've been messed up if you'll take your time and go back through the data. There'll be something there that will show you if you've been observing and writing things down. There'll be something that goes through. Sometimes though, you need to have a separate pair of eyes to look at it. I had Henry. He helped me out a lot of times because, you just feel like quitting. Anyhow, that's what I did. In life, when you're going through and it seems like what you think you want is not working, ask yourself, do you really want it? Do you really want this? Is this what you want? Is this, when you go to sleep and you wake up in the morning, is this what you think about doing? Will this make you happy? Or are you excited about the money that comes from this? Or are you excited about living up to someone else's expectation? Is that why you're going after this? If this is what you really want to do, it's time to come up with a strategy. It's time to come up with a plan to get to where there is somebody, they say you're 3 steps from 3 individuals from anybody in the world. There's somebody that knows somebody, right? There's somebody who knows somebody who knows somebody. So once you come up with your strategy, start talking to your network. Start talking to people and seeing who can help you get to where you need to go. Don't be ashamed to ask for help. That gets back to that good help. Don't be ashamed to say, I've gotten here, but now I need your help. PhD doesn't mean perfect. It just means we went through and we said we didn't quit. We're still human. So ask for help. Seek someone else to help you out with your career goals. And I promise you, you'll get there. I'm trying to be not so wordy, Henry.
Yeah, thank you. Thank you for that. I think it's important when you say seek help because a lot of people don't want to reach out because they feel like that person would not, email them back or, contact them back. One of the things I've learned over the past year, especially in 2020, when we've had a lot of downtime, is that, you might send out 40 emails and only get back 2, but it only takes 1 to get you the opportunity, right? And so, I'm following up to that. I think it's important for people to actually reach out to people. If you want to be, someone who's involved in regulatory affairs or science policy, find somebody who has that job or has that position, right? Reach out to them to learn their path. Not saying, hey, hit them up, I need a job, but saying, hey, can I learn from your journey? Can I learn from the mistakes or, all of the achievements you made in life so I can take that and apply it to my own journey, or try to implement some of the things that you've done so that I can get to where you are right now. So I think it's important when you're approaching people, you're not approaching them for a favor, you're approaching them for education, to learn. I think that's really important. And thank you so much for getting that. I think we're running out of questions and we're coming up on time, but I think one last question is, what's 3 attributes that you believe that we would need to continue the fight against cancer, right? We are in an area where we are the minority affected by cancer the most, but we are also the minority who are on the front lines working against it, right? So what are 3 attributes that you think we would need to continue this fight?
Whew, I would love to have, this is the whole, that question by itself could take up like a half hour.
Okay, we're not gonna do that.
No, I'm going to give you it very quickly. Don't worry. But I think that it's important that we're inquisitive. Remain inquisitive. Remain inquisitive in that you're looking out, understanding that there's always something going on. So as you're seeing in the news, as you're reading new papers, ask questions. Learn to develop questions from what you're seeing and what you're hearing. Why is this, for example, they have the Pfizer vaccine and they have the Moderna vaccine. Why is one having more, seemingly having more side effects than the other? This brings about a branch of study or somewhere we can look into. So be inquisitive, but in your level of inquisition, remain teachable. So you have to be always ready to receive information from others. And it's not always about someone up, meaning someone who's been there longer and someone who's been there, who's got more degrees or letters behind their name than you, but be open to receive and teachable from everybody. You can learn from any and everyone if you're willing and remain teachable. So be inquisitive, remain teachable, and be hungry. Be hungry. The problem is once we get to a certain level, I think we feel like we've achieved. Don't ever feel like you've achieved. Always recognize there's a little bit more you can go for. Be happy where you are, but be hungry to go for a little bit further. It's the early bird that's hungry that gets that worm, all right? So I took 30 minutes and wrapped it up into hopefully 30 seconds or so.
Thank you. I think those are all the good things we can all learn from all of those traits. Those are things that we need to, take in, for science and outside in everyday life. So I really appreciate all of that. I think we're going to bring it into time, but we really wanted to, thank all the speakers earlier for communicating their science, because it's really important for our community to see us, right, in this space, talking about the things we do every day to help our community. And it's also amazing that we could hear from you, Kelvin, on a different perspective of how do we use what we learn every day in lab, right? How do we apply that to everyday life, considering all the things that's going on in the United States and over the world. This is a really strange time. And I think it's important for us to have community sense, which is why Black in Cancer was formed, so that we can have a community to reach on and lean on for things like this. And so, I really want to thank everyone for joining in. We're coming to a close, but I really want to thank everyone for taking the time out of your day, because everyone's busy, and this 2-hour slide, you could be running an experiment or something like that. So thank you for taking the time to come and listen, because it's important that not only do we pass this knowledge on, but we're able to apply this to, our everyday lives. So thank you again. Also, one little quick, Kelvin, everybody wants to know if you have a podcast because they're ready to subscribe.
I'm working on it. I've got my page. I've got a website that's coming up, Dr. Kelvin Jones. It's coming up, and I'm working on a podcast. I'm working on it. You can find me on Twitter at, oh God, what's my Twitter handle? Henry, what's my Twitter handle?
KMJ. I'll tweet it out. You're not really active on Twitter, but I think now you have to be.
I'm working on it. I have to be.
Now you have to be, because you have all these people wanting to learn from you. I think that's, we could use your tweets on a day like when we're failing in lab or we feel like a failure. But everybody needs that podcast, so let's get on that.
Will do.
But if there's any questions, you can always direct them to blackincancer at gmail.com, or you can feel free to DM us on Twitter. We're really active there. But thank you again, everyone. And I hope you have a good rest of the day.